dynamic surge. This is where the Department of Wildlife, Fish, and Conservation Biology is. And that's where John Eady's chair is, and the Museum of Fish and Wildlife and the Entomology Museum is housed. So, and also where my office is. So we're probably gonna go talk to Andy Engilis, who's a curator at the museum, and he'll show us some cool stuff about all the different specimens that we get to work with here, and the university's influence with that museum, and other aspects of the U.S. here, and then also the neat waterfowl history and heritage that kind of helped the museum come about.
Hi, my name is Andy Engilis, and I'm the curator of the Museum of Wildlife and Fish Biology here at the University of California, Davis. And I want to welcome you to the collection room that houses our bird and mammal specimens here at the museum. So in this room, inside these cases, are about 30,000 specimens of birds and mammals. And these specimens are used for primarily teaching and research. We support almost every class taught in the Department of Wildlife, Fish, and Conservation Biology with our specimens. Some of the classes, like ornithology or waterfowl ecology, are courses that use our specimens intimately as part of their instruction. But we also support ongoing research by ourselves, as well as by our colleagues here in the department, and even broader outside our university. So the museum is basically a resource that can be used by a lot of researchers looking at evolutionary questions, or plumage, molt, conservation, lots of questions that require sometimes looking at specimens. And so that's why we're here. And our job is to maintain the collections for those use. Some of the oldest specimens we have in our collection are from the late 1800s, but in all intents and purposes, this museum started in 1972. And so it is one of the younger museums in the United States, but it has become one of the largest university collections in California. And it is now nationally and internationally recognized for the holdings that we have in support of research. So just to kind of show you, I'll start off maybe with Hawaiian duck. This is a case of specimens of Hawaiian duck, and these cases are specially designed to protect the specimens from the elements. So looking at dust, we want to minimize dust. Light's a really degrades feathers and fur, and so these cases helps support those and to minimize those issues. So this is a drawer, a typical drawer. If you open up any drawer in our collection, whether it was a mammal or bird, this is the way that specimens are organized. For birds, we do what are called museum round skins. And so this is a typical museum specimen. A lot of people think that museums have just animals that are live posed. And you would think about how many cases would you need if you had all of these ducks posed flying or mounted standing up. We would need a lot of space. And so museums have, over the centuries, devised the study skin. And so this is a typical study skin that we prepare. Not only do our professionals prepare it, but we teach undergraduates how to prepare specimens. And so a lot of these specimens you're going to be seeing today have been prepared by undergraduates that we trained. We also do flat wing mounts. So this is a wing of a Hawaiian duck, and for anybody that knows mallard, you can see there's a lot more green in the wing of a Hawaiian duck. It's hard to get that, but there's a lot more green in their speculum than a mallard. Mallard's more purpley to blue, and they're a smaller duck than a mallard. So the wings are really important because we can actually look at aging birds with wings, and whether it's a duck or a house sparrow, you can age birds really well with what's going on in the molt of the wings. So we always keep wings of every species of bird. So what's cool about the Hawaiian duck is that I was in the 1990s, I was a biologist working for Ducks Unlimited in the Western Regional Office, and I came to Ducks Unlimited from Hawaii, where I worked on the conservation of water birds in Hawaii. And one of the endangered species in Hawaii is the Hawaiian duck. And so when I got to Ducks Unlimited, we began habitat work for Hawaiian duck. I had a master student who worked on habitat, modeling habitat, and then we got involved in a study looking at the genetics of the bird in relation to its hybridization with mallards. So mallards were introduced to Hawaii, and they hybridized with Hawaiian ducks, and it was a threat towards the conservation of that species. So, you know, we would get a bird like this and think, oh my God, this bird is a hybrid. This is a hybrid mallard Hawaiian duck because it's showing all these traits of mallard. And what we found out is that this actually represents an immature male Hawaiian duck. And the genetics of it is pretty interesting, but there are genes that are contributed by mallards to the Hawaiian duck that turn on and off based on hormones. And when they're turned off, the bird will be brown, and when they're turned on, you can get different levels of trait expression from mallards. So like these are two pure Hawaiian ducks, but they show differences in the amount of gray. There's a little white, you have the little white ring around the neck on that bird. And so every bird, every young Hawaiian duck is different. And we couldn't figure it out. We're like, well, what's going on? And then we looked at the genetics of the bird, and we did full genome sequencing, working with colleagues with Phil Labretsky at University of Texas El Paso and John Eady here at Davis and myself. And we began to look at the traits related to the genetics of the bird, and found that these markers, these genes were there, these mallard genes were there. So what happens is on this bird is that when it's young and there's not a lot of testosterone being developed, it will trait express mallard traits, and as it gets older and has more testosterone, the testosterone is metabolized into estrogens, and the bird basically matures and becomes brown. So it's very female-like because the hormones are just really always in this bird's body. It's a tropical duck, so it's always ready to reproduce. And so it's basically got this sort of brown plumage as a result of that. So if you were to have a situation where the testes would get smaller, like you have in a North American mallard, there would be times of the year where this bird would get brighter, and then times of the year where it would stay brown. But in this case, it's brown year-round, and that's the condition related to the hormones and the breeding, year-round breeding of Hawaiian duck. So that just is an example of how specimens have been used to correlate. So we've been able to look at plumages. We now have, because of the genetics that we've done, all of these birds have been genetically vetted, all these Hawaiian ducks, and these all represent different plumages. We can look at, again, molt, timing of molt, trait expression, and we can correlate all of that. And if you were going to, for example, remove mallards and hybrids from Hawaii as a conservation measure, you're going to have to be careful, because you have pure Hawaiian ducks that show what we would consider hybrid traits, but they're not hybrid. This is a pure Hawaiian duck. So,
and so is that similar, like, kind of, you know, we've chatted before, but as a duck hunter, sometimes I've harvested ducks that either look kind of like a drake but have hen characteristics versus a hen that may have the green stripe, and sometimes people say, oh, a black duck mallard hybrid is, you know, that some of the testosterone estrogen stuff similar, and how the expression of male and female mallards look.
So for monochromatic ducks, so the black duck, Mexican duck, mottled duck, you know, all those species, there's part of the genome that's contributed by mallards, and so that is what's causing some of the variation that's seen. But I think if you want to get at the question you're asking at, let's look at northern pintail.
All right.
So, because let's get out of the mallard complex and look at something where we can see. Yeah.
That's the Arkansas in me.
I just have to find it real quick here. And a cicuta. I knew there was right in this area here. Okay, so the timing of when you're hunting is obviously going to be really, really important. So these are specimens of, I'm going to put my glasses on here. These are specimens of northern pintail. This is one of the older specimens we have here. This is a specimen. This was collected in 1975. And we probably have some that date back to the early 1900s. But at any rate, this is what, everybody would consider, this is the sprig, and this is what a lot of hunters are trying to get. But if you're hunting early in the season, like say openers, like oak, in October, or in areas where maybe the habitat's not quite right because of drought or something like that, and the birds aren't molting normally, you can get various plumages that occur in the population. So let's see if we can find. So here's a really good example. So I mean, this is a drake. This is a drake pintail in what we would consider its eclipse plumage. The technical term would be the alternate plumage, and this bird is already starting to molt. It hasn't quite finished, so it starts looking kind of female and male-like. But one of the things that helps you identify drakes in this plumage is that the soft body color parts, they still express male traits. So the bill should be black with the big, blue side saddles. That fades in a specimen. The other thing too is that because of the way ducks molt, they molt their flight feathers first, and they grow their flight feathers back, and then they'll fly to an area and they'll do their body molt. So this bird is already got its fresh adult male wing. I mean, this is an adult male wing for sure. I mean, these broad secondary coverts, and the speculum is adult male. So we know this bird's an adult, so we know this bird is now in what we would call pre-basic molt. So it's going to come into its basic plumage later in the season. So what's interesting about this bird is this is a 21 November bird. So if you shot this bird, you might think it's a hybrid. Maybe it's a mallard pintail hybrid, but it's one of these birds, and the year that it was collected was 2010. This was 2010. There may have been conditions that caused this bird to stop molting. It suppressed its molt, because usually by November these guys are in their full plumage. So we can look at, you see variation in those types of plumages. Here's another bird that's kind of similar. This bird is from October, so this is like the first week, the opener, probably 1972, October 23rd. So this is another drake pintail that was collected in its eclipse plumage.
Yeah, it's interesting to know, because I how the wing gets the more mature plumage first, because it makes me think of, kind of like with the parts surveys, so they ask you to send those wings in. So sometimes you buy a hunting license, you get asked to keep some of your wings in the manila envelopes to go to the wing bees, so you can get those estimates. So it makes you think how important, because I've gotten ducks before where you look and you're like, wow, that wing looks like, this species, this sex, and then you look at the body and you're like, wow, this looks a little off. So yeah, it's interesting to know.
So like if you look at a lot of collections, I mean, when would we most likely get specimens of ducks? And the answer is during hunting season. And so any museum you go to is going to be biased. It's going to have specimens from October to January, and they're going to have this bias unless, like in the case of California Department of Fish and Wildlife, we have a permit that allows us to go out at any time of the year to collect a bird, so that we can show and define the other plumages that these birds go through. So these are female birds right here, and again, you can age and sex them differently. That's actually, there's a whole lab in one of our waterfowl ecology lab on aging birds by their wings. So that's just an example of, you can get a bird that's anywhere from almost completely brown to, a beautiful drake sprig. And then, there are hybrids that occur. But there's also another plumage trait. Let's go look at mallard real quick. That's kind of cool. Let's go over here so I can find that bird quickly. So here's nice, big greenhead mallards. And interestingly enough, really been trying to get a lot of modern specimens of mallards, because a lot of our material, you know, they're so common that we don't take them very often. And so in the like 2015 on, I've been trying to get more and more modern ducks, and I talk a little bit about that. But you can tell this bird right here is kind of cool, because this bird has been sampled for a research project by Dr. Lavretsky at University of Texas, El Paso. And so every specimen that has had some kind of sampling of it, we put a tag on it to basically say, look, that's what's going on. So this is actually a project where he's doing full sequencing, full genome sequencing. And he has a grad student coming out in September to measure all the birds that he sampled.
Cool.
In our collection, as well as other collections around the western U.S. So if we look at, so then we have molting mallards. I mean, this is a Drake mallard right here, in full, we would call this full eclipse plumage. And this bird started its pre-basic molt because it's dropped all of its flight feathers. And so it's going to change out all these feathers first, and then once it's done doing that, then it will change out its other feathers. So that's like as deep a molt as you. This is like as deep. This is as far away as you could get from a greenhead, right?
Yeah.
And you'd be unlikely to encounter this plumage hunting
season. Yeah.
But you might encounter them transitioning like this. This bird's starting to transition. So you can see it's starting to get the chestnut and stuff in. So these are really important. You know, you look at any wing bee or any like wing manuals or any, I don't know, it focuses on immature birds of the year or adult birds. And it doesn't focus on the breeding birds at all. Yeah, so trying to find, for example, try to find what a female wing looks like, an alternate female who's on a nest, is difficult. It's hard to find.
And that's what a lot of the wing bee training I've had. It's all on wintering birds, and some of those rules go out the window when you get to the springtime, because it changes wildly. Right.
So then what we do is, for each duck species, in particular, we have a really great waterfowl collection. The heritage goes back to Dennis Raveling, who was the first waterfowl ecologist here at UC Davis, and he wanted to establish every species of North American waterfowl in our collection. And Dr. John Eady has sort of continued that legacy. And then with my interest in waterfowl, we've really pushed the limits on trying to even get some of the non-California species in particular. So we try to get different age classes of all the different ducks. So this is a great drawer to show you. Most of these youngsters were mortalities, birds that died that people picked up. They may be raised in captivity and died in captivity, but they represent the aging process of a mallard up through it's becoming almost a juvenile mallard. I wanted to show you so this is kind of a cool drawer here. The specimen is really super cool. So here's, do you see what's wrong with this bird?
It's got orange beak, green stripe.
yeah. So this is a female. This is a female mallard, and this is it's called an androgenic bird. So this bird has, as mallards grow older, which is rare, female mallards grow older, which is rare, they actually get more testosterone in their bodies and less estrogens, and actually the estrogen levels just drop. And so there's like this balance that's going on, and there's just not a whole lot of hormones in this bird, and that switches on those male mallard genes that express traits, so you can get this variability. But again, the soft body parts stay the same. So this is clearly a female bill.
Right.
And so how do we confirm it? The way we confirm it is we look inside. So when we get a specimen and we take the body out, we open it up and look for the gonads, and yeah, it is definitely a female.
Right.
So this is a female androgenic female mallard. So probably a really old
Right.
Old female.
And so, do you know exactly what old is in a female mallard, in terms of like how many years a mallard might take till it starts to express that, if it was to live long?
Off the top of my head, no.
Oh, okay.
So with Hawaiian ducks and Laysan ducks, when they get old, they get white. It's kind of like almost like humans. So when I started as a biologist, I had black hair, but I don't anymore. So here's an example. This is Laysan duck. So this is another endemic island species, and this is a male. This is a young male, probably a first-year male, and this is an old male. And as they get older, they get more white on the head. And so I know John's been working on the eye patch of females.
We talked a little bit about that with with the eye patch on the female. So we're measuring that, and typically in an older female, you'll see the bigger one from what we've been observationally seeing. But
so in the case of Laysan duck, the white is related to age, and we don't know if that's like a social signaling. Or I don't know if anybody knows if there's a mechanism involved, or if it's just aging feathers, aging follicles, or something like that. But this is a pretty cool bird, and again, we have one of the more definitive collections of Hawaiian duck. You can look at you can see how fresh these feathers are. So this bird's molting right here. It's all patchy, and this is a really fresh bird. So again, we can look at these types of things with skins. You look at how worn. Look at the tertials on this. How worn they are.
We were seeing that in some of the wood ducks there.
Yeah, that's the time of year you would see that. Right now. So those woodies, they should be going. The females are already in their alternate plumage, but the males should be all female-like now.
Right.
Yeah.
Actually, I've seen recently, a couple days ago, there was a male acting looking wood duck on the creek, but it looked completely female swimming around out there.
Yeah. Okay, so let's see here. So we'll go down to this. We'll go down real quickly to the sea duck. So Raveling had at one time several graduate students working in Alaska, and one of the things that they were charged to do was to basically begin to get specimens of the eiders, the various species that they were working on up there. So these are king eiders here. There's female, and there should be these are common eiders. Let's pick up this drawer here. So spectacle eidred was one that they worked on. This is a really spectacular bird, and it's also a bird of concern. Its populations have really crashed. And again, you can see we have these different age classes here of them. Again, students, researchers working where they would find these dead babies, and then they preserve them in the field up there. They actually stuff them up there. And so this is a nice breeding adult, and then here's females from that. So our collection is pretty complete. If anybody wanted to come and work on molt and plumage or geographic variation, this collection's one that people would come to. And so that's kind of one of the importance of having complete collections of birds. So not only do we want it, so oftentimes we'll just take what we can get. Anytime an eider comes in, we'll add it to the collection because it gives us maybe a new year, a new location, maybe a new plumage. All of these earlier specimens, there are no tissues for them. So our policy now is if we prepare a specimen, we keep liver, heart, and muscle. And those are archived in our tissue collection. And that tissue collection is used more than any other part of our collection. Researchers doing genomic research. And so we have multiple researchers right now, looking at small mammals, looking at birds, that are utilizing the collection we have, and fish even. Fish.
That's awesome.
So that kind of that's kind of the we're working right now and trying to get some eiders in from Greenland. This is the Steller's eider. And this is a really different looking bird, and Phil has a new paper out that came out on this bird, and there's evidence that this species of eider evolved from a hybrid event between long-tailed duck and
I can see it.
one of the other bigger eiders.
Yeah.
And so now Phil's looking at the Labrador duck.
Oh yeah.
To find out if there's any hybrid, issue. Labrador duck's, of course, extinct.
Yeah,
It's
I
extinct.
assume is that Smithsonian collaboration there?
Yeah, Smithsonian collaboration.
Wow.
Yeah. So
Those are cool duck.
yeah, so these are kind of neat birds. All right, so I'll show you a couple of other quick things, just because we're more than ducks. Yeah, so I'm going to show you a giant Canada goose. So that subspecies Maxima, a lot of people think it's extinct. It's been hybridized out, but Dennis Raveling worked on these, and so we were able to obtain a couple of what probably are among the last of the giants. And when you see one, you'll never forget it. Okay?
Wow.
When you see a giant, you'll never forget it. So here, I'll let you hold that. So that's a giant. Look at the neck on it.
Holy cow.
And I like it when they say, "Oh, I shot myself a 17-pound goose," or whatever.
And is that a band that was put on by?
Yeah,
an
this
agency?
is probably it's a lab reared. It's a bird that was at the Northern Prairie Wildlife Research Center, which is where Rav was. So in the western U.S., well, in the eastern U.S., this is kind of like your typical the lesser Canada geese. This is more typical.
Totally different.
Yeah, and then Moffat-eye, those are the big ones we have, right?
Right.
So let me show you that compared to the giant.
Wow.
Let's look at that real quick. I think it's
a lot of goose.
Yeah, I mean that's the size of a goose.
Yeah.
I mean a swan. That's the size of a swan. Yeah. So Moffat-eye is what we consider. It's called the Western Canada goose, and it's the big goose here. And they're pretty big goose. So I don't know if we can
turn him around
here. So it's comparable to the giant, but not quite as big as the giant.
That's wild. I mean, even if you look at the feet there, feet are massive.
So these were found around the Great Lakes, I think,
wow.
historically, naturally. But this is the big goose that now has been introduced throughout the western United States. I'm not sure if they've been introduced to the east. But isn't that cool? That's
imagine having a bunch of those flying into some decoys. It'd be huge.
I mean, minima, like you want to see variation in Canada geese. I'll pull this over. Try and grab a cackler here.
Oh yeah. Small ones. Wow.
That's a mallard-sized goose. Is a cackler. This is probably, this is a minima. This is from Yolo County. Dennis Raveling collected this bird, actually.
Wow.
19.
Tiny goose.
Yeah, no date, Dennis. Tisk tisk. Bad scientist. Anyway, yeah, isn't that something?
That is wild.
That's something else. Anyway.
That is incredible.
So this is collected right outside of Davis. So now you can say you've seen a giant Canada goose.
And it is in fact giant.
Yeah.
It's as giant as you're going to get.
Yeah. Sandhill cranes.
Oh yeah.
And, again, one of the projects, a lot of our projects, when I was a biologist in the western U.S., a lot of our projects were geared towards conservation for cranes as well as waterfowl. So roosting areas, they tend to roost in shallow wetland habitats. And so the ponds that were being utilized by waterfowl were being used by cranes for roosting sites. These are different species from different eras. Let's see if I can. Here's again a wing. Yeah. This is the wing of a sandhill crane. And again, we try for every species. We try to have these wings available for people looking at mold and so forth. So a lot of museums are doing this now. A lot of museums are. We didn't invent the wheel. We're kind of following the trend of collecting wings on birds. And so we've done. Yeah, look at that. And then these are some whooping cranes. These are. This is a critically endangered bird. I think DU's been involved in some conservation with whooping crane. These birds came to us from the Patuxent Research Lab,
Okay.
where they had a captive population, and these are more, birds that died in captivity. And I negotiated with them for about 3 years, and they finally said, "Okay, just so you'll stop pestering me, we'll give you a few birds." And so we got some.
And the Patuxent Lab's the bird banding laboratory that is kind of in charge of all of our, when we call in a duck band, they hold on to all that data that people can extract.
Right.
Yeah.
Right.
Cool.
Yeah, that is exactly right. So anyway, so there's just, there's a lot more going on. And so in 2005, I started working along the Texas-Mexico border, just doing general bird inventories, specimen-based. So we go out and we collect birds and get the tissues into the tissue collection. And, it's resulted now in us being able to compare what the diversity is like now versus what the diversity was like back in the late 1800s, when the Smithsonian had bird collectors along the border collecting birds. And so it's really super cool to be able to look at the avifauna in an area, the diversity in an area in today's era related to how it was 125 or 130 years ago. Unfortunately, in California, there are a few areas that were neglected. And one of the major areas that was neglected was the Sacramento Valley. And it's like this big black hole of knowledge, of historical knowledge that we have about bird life of the Sacramento Valley. There are not very many specimens historically. Joseph Grinnell at UC Berkeley, who set up the Grinnell transects that across California in various areas, just said, "I don't want to work in the Sacramento Valley. It's flooded, it's full of mosquitoes, it's already been altered, no." And so they didn't work there. And so after we finished our work in Texas, I met Paul Bonderson at that time, and he's was president of DU at that time. And he and I got together, and he says, "Why aren't you working in California?" And I said, "Well, he says, "If you could work, where would you work?" And I said, "The Sacramento Valley." And he says, "Come see my property," and that's Birdhaven Ranch. And so in 2017, we got up to Birdhaven Ranch, and that was the kind of the genesis of the Sacramento Valley Bird Survey Project. So I pulled some specimens out so we can kind of see what that means. So when you do an avian inventory, a museum-based, specimen-based avian inventory, we look at trying to document every species of bird by specimens. And in the case of Sacramento Valley, we have multiple counties. So we have Sacramento, Yolo, Glen, Butte, Colusa, all these different counties. And so we're looking not only at trying to document the species, but also its geography. So oftentimes you'll see a tray like this, and you think, wow, that's a lot of flickers. Well, they're flickers from different years, from different locations, and different times of the year. It could be breeding birds, it could be wintering birds. And so this just is an example, a snapshot of just some of the diversity that we currently have documented in Glenn County. And a lot of these birds were collected at Birdhaven Ranch. Not all of them, but a lot of them. Like this is a interesting bird. We never recorded this bird on Birdhaven Ranch until 2022. And this is called the Lewis's woodpecker. And they're a bird that erupts. They erupt every so often into California in big numbers. Like, I don't know, they're coming from the Great Basin, I guess. And they just come in by the thousands, and they're all over. And there just happened to be some on Birdhaven that particular winter. This was January 14th. And so, again, now we've documented this bird, that we've got tissues so people can look at the genomics of it, and genetics of it. And then also we'll publish on it. So it'll show, we'll be able to create this publication that will document all this diversity. And we pay that forward to future scientists. So what I'm doing right now with this is, to me, it's important because I know the effort that goes into it. But the actual value of this collection, the Sacramento Valley collection that we're doing right now, is going to be how scientists can utilize these specimens and the tissues
and the information we have 50 years from now when they look back and they're looking at maybe tracking climate change or tracking habitat change or water management change or whatever. And so there are some researchers that have articulated and published, including myself, that paying specimens forward is one of the most important things we can do for future researchers because we have the actual organism. So when I first started collecting birds in 1981, nobody was collecting specimens for genetics.
Yeah.
Right? It was just, we were collecting birds, and for this in this museum, we were collecting for teaching. And in 1986, I participated on one of the first expeditions to New Guinea where we took liquid nitrogen dewar into the field and collected tissues from birds in 86.
Wow.
And so now there are scientists that will come to this bird and say, I'm going to pluck some feathers from the chest of this bird, and we're going to look at the diet of this bird.
Right.
Right?
Yeah.
Or they can sample a toe pad and take a clip of tissue, and they can look at the genetics.
Right.
And they can look at population structure. They can look at gene flow. And I'm only imagining what could be done 50 years from now.
Right.
So if a person has, that like Lewis's woodpecker, and they want to figure out where, I bet you 50 years from now they could sample that bird and tell you where
exactly
what tree came. I mean,
that's amazing.
Who knows what's going to happen? So there's been a renaissance in the use of specimens in museums because of the technologies now that have advanced where they can amplify DNA from specimens.
Right.
And so again, the ultimate value of this is the information we have now, but the ultimate value is how it could be used 50 years from now.
Right.
And there's some really cool birds in here. Like this bird, I remember the day this bird came in. We were at Birdhaven Ranch and we had a mist net out. We used it to capture birds. And we captured this bird right here. Actually, Danielle found it, Danielle Fredette. She was an undergraduate working for me. And this is a wood thrush. And this is a bird that does not occur in California. If they occur, it's because they're lost. And so this may be one of the only specimens of wood thrush in any museum from the state of California. I don't know. There might be one here or there, but this bird was caught at Birdhaven Ranch at a fruity mulberry tree, which is just behind the slough house property there. And so it was, I think it was June. It was July 13th. And that's pretty unusual. So this bird was really lost. This bird really lost. More typically, we have, this is Swainson's thrush here. This is hermit thrush. More typically, we have these in migration, this species in winter, and this is just a vagrant, a lost bird. Here's another one. This is a mountain bluebird. And this was collected on a piece of property, not at Birdhaven, but near it in Glenn County. And this is a mountain nesting bird, but they do come down to the valley floor every so often to winter, and very, very few of them. So this is again, there are probably very few specimens of mountain bluebirds from this Sacramento Valley.
Right, right.
So that kind of utility is cool. From my perspective, I look at records and look at how they correlate to our modern perception. And then these are like different shorebirds from different times of the year. I wanted to take you over to this tray here because this is a super cool tray. This is a tray of birds that we have. All these birds were obtained in Sacramento County, and almost every one of these birds were picked up by a citizen who found the bird dead and were concerned about disease, so West Nile virus or whatever. And the Sacramento YOLO mosquito vector control program will take birds from citizens and test them for West Nile virus and now avian influenza. If the bird, and they knew about our project, and they said, well, do you want our birds? And I said, yeah, I want your birds. So every one of these birds were picked up by a citizen, found dead. Normally they would get tested and thrown out, burned. But because of our project, we now archive all of the specimens. We get about maybe 200 birds from mosquito abatement every year.
That's awesome.
Yeah, and so we have a really great working relationship with them in this county to allow them. I mean, there's some cool birds here. I mean, this little guy right here, this is a flammulated owl. And this, again, is another mountain breeding bird that's super rare on the valley floor. And citizens have picked up 2 of them during the course of our study. This one was from 2017, and this one was from 2020. Wow. And if that surveillance wasn't going on by the general public, we would never have ever obtained these.
Right.
So just to give you, this is a screech owl, so this is a more common owl in Sacramento area. But look at how much smaller the flammulated owl is. It's when they came in, they called it a screech owl.
Right.
And I said, that's the world's smallest screech owl. So it's really super cool.
And those would largely go undetected because it would be nighttime, right?
Yeah, they migrate at night. These guys migrate. They migrate at night. Yeah, we would not detect them. There are a few observations here and there, but again, this is a least tern. This is a bird that was they nested at the Sacramento wastewater treatment facility. And there were like 2 of them, a pair that nested year after year after year, and one of the pair died in I think it was just 2018. One of the pair died, and they found it and sent it to me, and I said, oh yeah, it's part of that. We can include that as part of our project. So we have an egg, an infertile egg, and we have an adult.
Amazing.
To document for our project.
Awesome.
Yeah. So, and then this is a Wimberl. We have, green herons and various kinds of hummingbirds and so forth. So what's really so cool about this is that you can get so much. Once we started plugging into the agencies that obtain specimens, we didn't have to collect very much.
That's awesome.
So initially we collected birds because we knew we wanted to get, certain species. But now, this project now has documented over 200 species, and almost 190 have been documented by specimen. Wow. So it's going to be a big monograph.
Wow. Yeah, it really shows you the importance of cross collaboration and also the amount of effort and hands that go into obtaining something like this.
Yeah. So beyond the teaching, this is the teaching lab that we're in right now. This is where we teach all of our organismal classes. So mammalogy, ornithology, ichthyology, and herpetology. Mm-hmm. And so we're one of the few universities left that still teaches all 4 organismal classes. And to me, it's fun. It's like learning how to read. Right. It's fundamental to becoming a wildlife biologist is to know what you're working with.
Right.
So those courses to me are just so fundamental to what we teach, and they're some of the most sought after courses by undergraduates.
Right.
So, anyway, so this is just a snapshot. We have more specimens than this, but this gives you some. These are different raptors. This is an osprey. This came from Birdhaven. This bird came. There was a nest. There used to be an osprey nest at Slough House.
Oh wow!
And this, I think, was maybe the male. I'm not 100% certain, and it just died. One day there were somebody. I think one of the people that work up at Birdhaven drove by, and they found it in the bushes underneath the nest. Oh, and ever since then, they stopped nesting.
Oh.
So, but we were able to salvage the specimen. So some of the specimens here have come from like Rancho Escuan too, mm-hmm. which is another hunting club up in Butte County.
Okay.
So we've had other clubs, donate their time or ability for us to go on the property and do.
Yeah.
So that kind of gives you a snapshot on the bird side of what we're doing, and just to broaden what the museum houses. Let's go take a look at the mammals real quick. Yes, you can just do that. You can see some of them right here. These are larger mammals, and our museum isn't really geared to be the world's greatest white rhino collection. But it's nice to have one because we teach in mammalogy. All of these skulls are used to teach family traits or order traits. This is a hippopotamus, and a lot of these came from local zoos.
And we have a standing agreement with the San Francisco Zoo. Whenever an animal dies, they contact 3 museums, and we're one of the 3. And whoever dibs it first gets it. It's all done. But this one came from the San Francisco Zoo, as did the giraffe. This is actually a wild shot rhino from 1960.
Wow.
It was donated to the museum by an older hunter. Walrus, that's a wild, probably found dead walrus skull. The tusks we keep locked away because they're ivory, and people steal ivory.
Wow.
So we keep the tusks hidden. We'll come over this way. I'll take a look at bats real quick, just because they're super cool. So our collection, one of the things that we're doing right now is we're working on, we have a research program and a research associate, his name is Alan Hitch, and he has gotten involved in zoonosis research, so looking at viral diseases that mammals carry, and we are providing sort of a zoological framework in the medical sampling. So doctors are going out and sampling bats because bats harbor a lot of the viruses that are concerning to humans right now, the coronavirus and things like that. Ebola, a lot of the mammals are reservoirs for the virus, and there's somehow cross-contamination that's occurring. So medical doctors are going out and sampling bats, but they haven't been doing it in a zoological framework. So we got involved with a group from Duke University in Singapore. It's a medical group there. And we formed this program where we have a zoologist, which is Alan, and he sets up the framework for how we'll survey. And then we collect voucher specimens, which most people don't do. So they're saying, well, we got it on this bat. Well, how do you know you got it on that bat? There's no voucher that says that you got it on that bat. And a lot of this work has been done in Southeast Asia. And so we've started to develop a really, really big Southeast Asian bat collection. And it's related directly to our research that is ongoing on viral disease discovery. So we were working on coronavirus 4 years before coronavirus, COVID-19 hit. You know, isolating, looking for, sampling animals for it. We didn't find any in any of these bats that we worked on. But that work in Southeast Asia now is wrapping up, and we have a project in Kazakhstan, in Central Asia, and we have a project in West Africa now that we're just getting going in West Africa. So like you can look at the diversity of bats. There's some really super cool bats. These are forest-dwelling bats. Actually, these bats I collected in New Guinea in the 1980s, early 1980s. So I did graduate work in this part of, here's my tag right here. You can see the 1984 in Morobe province. So this is some, most of my specimens that I collected in New Guinea are at the Bishop Museum in Hawaii, which is where I worked. But I donated a few to the museum here back in the day. I'm trying to find the Tropus here. And then bats get really big. So this is a fruit bat, people call them flying fox, but they're actually a fruit bat, and they don't echolocate. They eat fruit, so they use scent and eyesight to find fruit. So they have big eyes. So again, these are just shows you some of the examples. These come out. We have a couple of these in the collection. They come out for teaching purposes. Wow. We don't want anybody touching this because the preparator used arsenic to prepare it, to preserve it. So we established a mission statement in 2004. And in 2005 and in 2008, I got back-to-back NSF grants for the museum. Wow. And so those are major grants that put us on the national headline. And then we just grew. But the growth was related to the research that we're doing, exactly what you're saying. Wow. So what do we need to be done? We're supporting genomic research. We're supporting disease research. We're just supporting conservation and ecological research. And so you know, the museum now is multifaceted. You know, we have 9 employees, we have a postdoc, I have a collections manager, and so my job as curator is to administrate the museum, but also to provide that scientific direction. So that's a really important role that I play, and then I interface with the faculty in the department. Make sure I've got projects with almost every faculty I can. I just try to like engage the faculty in the museum. You know, make it a resource that is usable. And then the other aspect is get the undergrads in. This is like one of the best internship opportunities are with this museum, whether it's in the museum curating specimens or prepping specimens in our lab or going in the field and doing field work. I mean, a lot of that work we did on Birdhaven, you know, it was undergrads doing it. And so I really, really have emphasized our internship program, and it's now become a nationally recognized internship program.
That's awesome.
Irene gave a paper recently at the National Academy of Sciences on our program and how we engage undergraduates, and so it's becoming really well known and modeled now. So yeah.
That's really neat to know, especially like if anybody's watching about, you know, wanting to get involved in a career someday. Maybe it's working with specimens, or you know, have an interest in the research, but maybe wants to work with specimens, or maybe wants to do research at a university with waterfowl or anything of that nature, but wants to kind of look at what's previously existed, like really tapping into either the museum that might be on campus or considering UC Davis as a potential place to come and a potential collaborations. A lot
of students come to this major and they cite the museum as a reason that they want to come.
Awesome.
And so, you know, that's exactly right. I mean, we feel like we are an integral part, kind of I like to think of us as sort of like a hub for the department.
Right.
And so this, you know, if you were here like in October, there are undergrads coming in and out every day of the week, working all day long, you know, different ones. I think we have as many as 50 interns a year that we train, either in the museum or in the Nest Box project on Aputure Creek.
Right. Yeah, and I remember the biodiversity day being in here because I taught waterfowl for 4 hours, and there was actually a surprising amount of undergrads that actually said they were, after they spoke to me about ducks, they were thinking about going in that direction. And so, you know, I've seen it firsthand how the museum really cultivates and encourages that learning environment and that career building, relationship building environment. So.
If you're coming to Davis as in the wildlife department, if you're not seeking out internships to get experience in field work or whatever, you're not doing it right. You should be getting things. You should be getting involved. And and I tell that about, you know, the museum. I mean, if you are an undergraduate and you've done, let's say the wood duck nest box project, but you have been a preparator, specimen preparator for the museum, and you're going on to vet school or you're going on to wildlife, having that on your resume, that you've worked and handled birds and mammals and in a lab setting, and you've been trained in anatomy and trained in how to prepare specimens, and there's detail-oriented aspects to that work. You don't just come in and sit down and prepare. You've got to do it properly. And it puts you in the upper.
Carries a lot of weight.
It carries a lot of weight. It really does.
That's
it puts you in that upper, particularly, you know, if you're looking for seasonal jobs.
That's awesome.
Because they're looking for people that, you know, undergraduates. Do you have experience in handling birds? Or bats or whatever.
Well, and that's interesting because I feel like a lot of people think, oh, I have to have experience going out and capturing specific animals that may be difficult to capture, when in reality, a university museum like Davis may be able to give you that experience and that opportunity, because you're getting stuff shipped from all over, and opportunities to prepare things you may not even get inside the United States. So that's a pretty cool.
Yeah. So our long-term goal right now, my long-term goal is to try. We've been working on a new building that would house the museum and increase the department's capacity to teach, and, you know, to have more labs available, more teaching opportunity, more internships. And it all starts, unfortunately, with space. That's like always the big issue at any university is space, you know. And so we're getting capped out in our museum, and so we're looking at ways to try and find donors, and maybe there's some federal and state programs that we could tap. But we have actively now designs and everything ready to go. So we're ready to really try and push that. And I've been working on that for about a decade now, just baby steps.
Yeah.
But we've really got to start thinking about that.
Yeah.
So that's our future, is try and get a newer facility that would expand our capacity.
That's awesome.
Yeah.
Well, thank you, Andy, for showing us the museum. We really appreciate it. I think it gives a really good insight to people that are potentially, watching, in terms of whether you're a waterfowl hunter or an undergrad interested in wildlife research, about the resources of a museum on campus. I don't think we think of that immediately, or some of us may not even think that we might have a museum on campus. Specifically, even just the influence that Davis's museum has had, and then also just kind of the waterfowl heritage, which hits the home for a lot of us. So I think with that, we're going to go check out the aviary that the ED Lab maintains and uses, and talk to a couple of other grad students within the ED Lab about their research projects. So we'll go head out there right now.
Yeah, and when you, if you're interested in the museum, just Google MWFB, and you can get into our webpage, and then that'll show, we have all these projects all defined. It's a great website, and you can also link to our social media there. So thank you very much. Thanks a lot, John.
Yeah, thank you.
Take care.